Okay, I'll steal 2 minutes from George. It's my great pleasure to introduce Dr. George Perry from University of Texas, San Antonio. George is a distinguished professor, a chair professor in neurobiology, and also the dean of College of the Sciences over there. And he's a well-recognized researcher in the field of Alzheimer disease and neurodegeneration. And I think he had, over the many years of research, he studied almost every aspect of Alzheimer's disease, including amyloidosis, metal homeostasis, and oxidative stress. And some of those topics he will talk today. Dr. Perry obtained his PhD from University of California, San Diego, and did postdoc at Baylor College of Medicine. And then he joined the faculty at Case Western Reserve University and rose, rising to the rank of professor and chair of the Department of Pathology over there before he moved to, or recruited to UT San Antonio as the dean. One of the amazing things Dr. Perry has accomplished is he is extremely prolific, published over 1,000 papers. And I remember, I was a graduate student at Case Western, and I have a number of friends actually work with George. And during like a third year or fourth year, you began to be very, more and more productive. We are celebrating our publication number 1, number 2, number 3, number 4, number 5. But when we talk, celebrate those, talk to his student, the number is like 10 times. One of the students showed me is published like 20 to 30. So we were totally amazed, and I still do not quite understand what's the magic in play over there. So I think I will highly encourage graduate students, have lunch with him, or we also have a reception at 3 o'clock, talk to Dr. Perry. What's the trick? What's the successful gradient to be so productive? Dr. Perry has held numerous, to be editor of numerous journals, including highly influential journal of Alzheimer disease, get very extremely highly cited. And he has a list of honors. I think I can only mention a few because of time. He's the fellow of American Association of Advanced for Science and fellow for Texas Academy of Science, and he's the past president of American Association of Neuropathologists. So without further ado, Dr. Perry, welcome.
Well, there's a number of great honors about being here, being the first in a new room, and hearing that I'm going to talk about tricks. I wish I knew a lot of tricks. And further about students, it reminds me that this week, actually, I have a MOOC, a massive online course that's available on the web, which is really set about the graduate student level or advanced undergraduates dealing with Alzheimer's disease under EDEX. And you can dabble in it. You can spend 10 minutes and get bored of listening to me, or you can do the whole course. And there's no charge unless you want a certificate, which I don't think has a lot of value for most people. Well, I mean, unless you need to be certified that you did that as a remediation. But you can see different parts, and you can fast forward through me if you think I speak too slowly or something. What I'm going to talk about today is kind of an overview of various aspects. I called it mitochondria, but I'm going to talk about 4 different aspects, assuming I can stay on time, about the disease, about oxidative stress, about mitochondria, about cell cycle reentry, antioxidant activity, about amyloid, and I'm going to talk about oligomerization. These are all topics that I've been involved with over my career. The key element and I can talk later on about my trick about producing a lot of papers. It's not complicated. One of them is this. I work with a lot of people, and all the people I work with are much smarter than me. That's a key element to them working. If they can't outsmart me, then they shouldn't be with me. and that they should know more than me within 2 weeks of working with me. It's not that hard. In other words, they're supposed to teach me. But I can talk more in detail if you have questions about it. Okay, so I have a group now that I work with in San Antonio. I still work with people that are, well, actually, Yonggang Li has now moved up to here, but a group in Cleveland I work with, and then people throughout the world. So first thing is, I've been studying oxidative stress when I was at UC San Diego at Scripps Institution of Oceanography. That's my first studies were in oxidative stress. So that means I've been studying for essentially 40 years, and I can tell you I don't really understand oxidative stress very well. I think it's really complex. And the reason I don't understand it very well is right here. This is where the field was in the 70s. And that is, I call it the rusty nail idea. It's something that chemists, always to pick on chemists, they would have this idea as opposed to a biologist. See, I'm going to be doing a few digs. It's the classic definition, and it's like thinking of a shiny nail. That we're meant as a shiny nail, and as time goes on, we rust, and our body fights that rust, and we rust away. And that's the idea that oxidative stress is something you're always resisting, and it causes stress, and that's what's going on. Well, that's why this is a non-biological idea. It doesn't deal with the biology of how cells and organisms deal with something. Because when they don't deal with something, and it rusts them, that means it goes to a non-homeostatic situation. And non-homeostasis has a consequence. It's called death. You know, the organism fails. And that you pretty much notice. So in chronic conditions, so this might be good for acute conditions. You know, if you have a stroke, you have oxidative stress, and you either get better or you die. But if you're dealing with Alzheimer's disease or aging, it's not a very good definition, because what's happened instead is the system's always in balance. And what happens instead is you have these responses, the antioxidant stress responses that bring the system back. And actually, if it was so simple as the first definition, taking a whole bunch of vitamin C and vitamin E would be a benefit. And it isn't. It actually screws things up worse. Okay. So why I'm showing that piece, I'm not going to talk about it. We've described every every type of oxidative damage you can find. You find increased in Alzheimer's disease. Every type of stress response that you can imagine, you can find in Alzheimer's disease. So kind of, well, what is this telling you? So this is a summary of those slides, just a very, very superficial summary. If you look at oxidative damage, dealing with sugars, protein-free carbonyls, hydroxynonenal lipid peroxidation adducts, or nucleic acid oxidation, which is mostly RNA oxidation. Initially, we did these studies thinking, oh, there'll probably be mostly plaques and tangles that are going to have these changes. They sit around in the brain for 20 years. They're going to be bombarded. It's going to change their chemical properties. And that's actually where we started in the mid-90s. And what we did is we described the tangles within neurons. So these are neurons in somebody with Alzheimer's disease. Well, starting as a normal neuron, assuming everybody starts normal. And then it eventually develops tau changes. And actually, the neuron eventually dies, and they go in the extracellular space. So I've drawn this as a timeline that we actually never look at. We only look at static pictures of people that die in disease. And most of my studies are done in humans and comparative with cells. So when we did this, initially we looked at glycation, and we found it was predominant within the filaments of the disease. But as we started looking at changes that occur much faster and don't require complex chemistry, like nucleic acid oxidation or protein oxidation by itself, we started seeing that oxidative damage involved neurons that were at risk of the disease, that had no pathology. And that's why I have drawn here this neuron, which comes over here, that has nothing abnormal in it, no tau changes, and yet it has oxidative abnormalities within it, of all the categories I talked about. And then we tried looking, well, what's abnormal here? You don't get oxidative damage without having some kind of abnormality, usually catalyzed by iron and copper, but also dealing with what is the contribution of amyloid beta, inflammation, mitochondria, etc. Let's see. There. So we initially focused on all the features I showed there, glycation, inflammation, etc. But our studies quickly moved to focusing on mitochondria. And why we did that is the large neurons that are affected by oxidative abnormality are very highly metabolic. And there have been prior studies of John Bloss and others saying that there were fundamental mitochondrial abnormalities during the disease. And as you know from your biochemistry classes, mitochondria are the major site of energy production for cells. You're squinting, thinking about, oh gee, Krebs cycle and all that other stuff that you had to learn about. Anyway, it's a major site of ATP production. It produces key metabolites. For Alzheimer's disease, acetylcholine is basically a metabolite. The neurotransmitter that's affected during the disease is a metabolite of mitochondria. It's the major site of calcium buffering, which probably underlies a lot of the changes I'll be talking about later. It's a major site of free radicals. When you're eating food, it's oxidizing through your body. And it's using iron and copper to oxidize, which are reversibly reducing and being oxidized. And when it does that, just like a car is not 100% efficient, you're not 100% efficient. So you produce partially oxidized metabolites. And those partially oxidized metabolites, like superoxide, are the source of the primary oxidant causing oxidative stress. It also is the site of programmed cell death, apoptosis. So they're really at the center of things. And for a lot of process, people are interested in Alzheimer's disease. So we decided to look at this feature of whether Alzheimer's disease had mitochondria within the pyramidal neurons in the same way we looked at oxidative stress that I summarized there. One of the key things that we did was made things simple enough that even I could figure them out. That is, we turned looking at oxidative stress into stains, that we could look at very high resolution within a cell, rather than grinding everything up and averaging 100 different cell types. So we did the same thing for mitochondria. What we did is, we looked at a study that had been in mitochondria and muscle for mitochondrial disease, and they had described, as you know, mitochondria have their own DNA that codes several key enzymes. It's 16 kilobases, and it's organized like bacterial DNA, meaning that it doesn't have introns, etc. So most of the mitochondrial proteins are coded in the nucleus, but a few of the key ones are made in mitochondria DNA. But while mitochondria become abnormal, one of the things that happen is a deletion forms. So we made these probes to mitochondria that would recognize a deletion. And when we did this, we did find the deletion, and we confirmed that by PCR, but that wasn't the most striking finding. What we really found was this massive change in mitochondrial DNA. If you look at this is an aged person. This is somebody with Alzheimer's disease, and when you quantitate it, it's about a 3 to 4-fold in mitochondrial DNA. And when we looked at protein, we also saw an increase. In this case, we did cytochrome oxidase, immunoreactivity, and we saw about a 2 to 3-fold increase. You'll see it's a little bit different morphologically, and it's because of, it'll be more clear in the next few slides why there's a slight difference, but it's both increased. Yet when we looked at cytochrome oxidase activity, it was reduced about 20 or 30%, either by looking at it by enzyme homogenization, bulk approaches, or enzyme histochemistry. It was not increased. And that was kind of a paradox for us. So we wondered, how can you have mitochondrial DNA increase and mitochondrial enzyme activity, if anything, slightly decreased? So what we did is we actually looked at biopsy samples taken for diagnostic purposes, which used to be taken during the 60s through the 80s, early 80s. It was considered to be an appropriate way to diagnose Alzheimer's disease to take a biopsy. In other words, out of a living patient. And although these are samples from a living patient are not equivalent to rats because you don't perfuse people. That would not be considered ethical under any circumstance. So these samples, you can see things that look like mitochondria with the cristae intact and a double membrane. You can see them with cristae broken, and you can measure those. And you can also look at something that is called lipofuscin, and there'll be the vesicles here. And when you do that, you can count those and look at how much area they occupy. And normal mitochondria, if anything, are slightly reduced, statistically significant reduced. Those with broken cristae are not even any different. And this is one thing, it's really important when you do morphological studies not to just show pictures. It's important to do quantitation. Because you can, I think it's important to have your pictures reflect the quantitation, not the reverse. May take an order of magnitude more work to do so, but I think it's critical. And then these vesicles here, and it'll be clear why I bothered to do this. And you see that there's, if anything, a slight increase in Alzheimer's disease. The other piece we did is we looked where is the mitochondrial DNA. So the mitochondrial DNA in situ hybridization can be done at the light levels I showed earlier, or you can couple it to gold nanoparticles of 20 nanometers, which are here, and you can look where all these particles are. Well, these don't look very much like mitochondria because they aren't. They're autophagosomes. And you see all the gold particles there. Now, I didn't show here. There is mitochondrial DNA in mitochondria, not surprisingly, but there's a whole bunch of it in these autophagosomes, indicating increased mitophagy, or at least accumulation of it. And you can also see it present in the cytoplasm. We did the same thing for cytochrome oxidase activity, and it's present within the cytoplasm. So the wrong place. So this led to work, and if you heard about a year ago, Jean-Wei Zhu came here, and these are studies that really he pioneered. In fact, pioneered over, not my objection, but his better insight, because I went down a different path that was much less exciting. So mitochondria within a cell, when I learned about them, when I was a student, they were those little round particles that I showed you, little bean-like structures. But the name actually refers to thread in Greek, because cells can either have 1,000 mitochondria or they could have 1 mitochondria, and they could have the same amount of mitochondrial protein. So this is here about knocking out certain proteins, and you can see all this thread-like structure through a cell, or you can have all these round-like structure. And this is controlled by a process called fusion fission that you probably heard about, but wasn't present when I was a student, so I didn't focus on it like Jean-Wei did. I instead was down here and looking at the issue. Mitochondria also move through a cell, and a neuron is pretty unique in that it has these incredibly long projections. You have in your body neurons that are almost 3 feet long. They extend from the back of your bottom of your spine all the way down to your toe. And all those proteins have to get transported out there, and mitochondria have to get transported because most of them are made there. So there's they have to go up and they have to come back. And that's controlled by moving down microtubules. So to understand this more, I went and measured the number of microtubules within the neurons I did the mitochondria. And these are actually easy to do studies. Once you take the 5,000 micrographs and print them, it's very easy to have an undergraduate student measure them. That student who did this work now is a plastic surgeon. I think I drove him to that, but but so here's a neuron, a piece of a neuron cytoplasm, and you can see paired helical filaments and a piece of a neurofibrillary tangle, and you can actually see within it microtubules. It's essential those are biopsy samples because microtubules decompose really fast in the postmortem period. So we quantitated the number of microtubules within the neuron. We counted the length, we counted the number of cross-sections, we did it a couple different ways. And what we found is there's really a major diminution. So you can see of controls looking at pyramidal neurons, the type that are most affected by the disease. You can see they have a much higher density of microtubules versus those in somebody with Alzheimer's disease. And surprisingly, there's no effect of whether the neuron had a neurofibrillary tangle in it or not, just like the oxidative stress phenomena. Little effect. In fact, if anything, there's a reduction. And that's why this picture kind of dramatizes. The two things can occur together, but there's a clear diminution. When we tried creating model systems of that, though, we were unable to recapitulate. So I took cells and diminished their microtubules using the codicil, and I didn't get any changes that were like Alzheimer's disease. And this is why I say Dr. Zhu is much smarter than me, because he instead focused on this process, that the size of the mitochondria is just like transport is controlled by a whole intricate system. So is mitochondrial dynamics by this protein called dynamin-related protein 1 or dynamin-like protein 1. And you'll see me. I'm mainly going to focus on that protein because that's where we have the most data, at least for this presentation. There are a lot of other proteins that I'll show data for. Okay, so this protein controls mitochondria kind of squeezing out to become 2. And who cares if you have 1,000 mitochondria or 1 mitochondria if you get the job done? Well, there's a lot of reasons why this needs to be a controlled feature. One thing is, say you have in a cell a defective mitochondria. What does a defective mitochondria mean? Maybe a change in the DNA. Let's see. A change in the DNA, so it becomes defective and it needs to be fixed. One way you can do this is you can fuse together the mitochondria and they could fix that issue. Another way you could do it is you could actually cause this where the cells don't fuse and you get all these little guys, that some of which are really extremely abnormal, and you could get rid of them. And that's an important dynamic process. You want to be able to have repair and you want to be able to get rid of the garbage guys. Another part, which I'll show more data for, is that neurons having these long processes called axons and dendrites, you need to have mitochondria that can get out into them. And if you have a really large one, like one for the whole cell, how are you going to sort of move it down an axon? So we looked at some of the proteins, and this is in human tissue, human brain tissue. We looked at some of the proteins that are important in this. Dynamin-like 1 protein, which I told you I'll concentrate on. People ask me about OPA1, etc. Well, you can see that this protein is vastly reduced. It's about 75% reduction in dynamin-like protein in Alzheimer's disease versus control. Is it and then this loading control for actin, and is it everything's down? Well, you can see Fis1 actually is slightly up. So it isn't just everything down. The big advantage of the system that Zhu did is that you can study it in cells that I was studying, and but he again understood much better to follow up on observations I had made. And that is, I had looked at fibroblasts, olfactory neurons, which you could grow in culture, and I looked at fibroblasts from people with Alzheimer's disease. And these cells, when you grow them in culture, have 2 different organizations of mitochondria. They have a dispersed organization, and you can stain these since these are living cells that you grow in culture. They have a dispersed organization around the cytoplasm, or they can be clustered. And that clustering, remember, is kind of reminiscent of that mitochondrial DNA clustering you saw in Alzheimer's disease. So you can count how many of these cells have this collapsed morphology. It's not an impressive number. That's what I thought when I did these studies. I published that piece about thing, and I thought, boy, this isn't really as exciting as I should have found. But it is increased between Alzheimer's and cells from normal people. It's a rare phenomenon, about 5%, is where in Alzheimer's disease, about 20%. The other piece, you can also find out that the number of mitochondria is slightly different too, and the size is slightly different. So you have this clustering, larger mitochondria. He went back and analyzed the micrographs that I showed earlier, and also found out that mitochondria in the brain are about twice as large. Why I'm saying this, students can very often, Jean Wei was a student almost at this time, because he started on these studies. Students can spot things that professors miss because we start with preconceptions. And that's what's always great about working with students that have less preconceptions. Anyway, so we also, when we looked in the fibroblasts, we found that reduction in dynamin like 1 protein. Not a 75% reduction, and the results are not as striking as the brain. But the big advantage is the brain is dead. We can't do very much with it. We can only observe it. And here we have these cells that have that. And then when he modified the expression level of dynamin like 1 protein, so he focused on this 1 protein, which plays a critical role in mitochondrial size distribution. If you use that knockouts or inhibitory RNA, you can actually start increasing the number of cells that have that morphology. Yeah.
DMP1 encoded in the mitochondrial genome or in the
Nuclear. Yeah, the ones that are in the mitochondria are mostly actually respiratory enzymes. Okay. And we also overexpressed normal dynamin like 1 protein and found some rescuing. So this protein plays an important role in morphology. In experiments I'm not showing, we looked at some of the other proteins that play like OPA1, et cetera, and we found they play an important role in the energetics. This protein didn't play any role in changing ATP production or radical production, but it just changed the dynamics of size. In experiments, and we've gone way beyond this, we've taken the fibroblasts and converted them to iPS cells, and we've converted them to neurons, which I don't think I have included here. No. We've converted them to neurons, and we're looking at the mitochondrial dynamics in those. So why are we doing that? Fibroblasts are not neurons. They don't have similarly long process, and we'd like to be able to understand much more in a longer projection situation. I'd also like to be able to understand why is there a difference between Alzheimer's and control. But in the meantime, in studies we did at the time, we focused on how we could understand this in cells that were neuronal-like. And in this case, we used primary neurons from rat. And you can see in mito-red just marks where all the mitochondria are when they're functioning. And you can see that they extend down process. This just marks where all the neuron is, and you can see the red and the green. If you change dynamin like 1 protein, you'll see, and I think you can see without dimming, that the mitochondria are longer and there's bigger gaps. So it does play a role in it. There's a bunch of other proteins and they change other features. So what is it about the fibroblasts and the other things that cause these, why is dynamin like 1 changing? Or the other proteins. So to address this, we looked at a couple things. We looked at oxidative stress, and I won't show the data today. Just adding hydrogen peroxide will change things. But the most relevant, I think, for Alzheimer's disease is amyloid changes things. So if we take the cells and we transfect the cells, these are not the fibroblast studies, these are neuroblastoma actually, is that if we transfect the cells with amyloid precursor protein, the regular stuff, or ones with mutation, the so-called Swedish mutation. It's a nomenclature. They named the mutation after the country in which it was first described. And these mutations are limited to small families. You start to see much more mitochondrial abnormalities. So where you end up with normal control like 5%, as I said, this clustering or change in thing. If you put amyloid precursor protein, you move to 20. If you have it with mutation, you start getting to 50%. That effect appears to be completely dependent on production of amyloid beta, because if you put in a base inhibitor, you reduce that greatly. Further, and this is a slide that's really hard to see because of the dark background, if you look at it in correlation analysis, dependency on amyloid, you see a high correlation. In fact, all over 0.9. Amyloid production in the cells is correlated with this clustering phenomena. And then we also looked at whether in cells expressing directly, if we could measure how the dynamics of mitochondria, because you can actually do this in cells by bleaching a cell. So, in this case, bleaching doesn't turn it white, it turns it red. And if you take a control cell and you zap it there, it'll turn red, and then you watch the recovery period, and that is the recovery of it mixing together with other mitochondria. And you compare that with one. And it's about a 3-fold longer time period that it takes if a cell is overexpressing amyloid precursors. So it's clearly changing the dynamics. So this summarizes, there are a lot more publications and there's a lot more work to do. What we think is happening in Alzheimer disease is that in normal people, mitochondria are distributed, this is up here, the normal situation. The mitochondria are distributed around the cell where they're needed for energy. That means that a lot of them would be down axons because there's a lot of electrical activity and energetics there, and dendrites and et cetera. And during Alzheimer disease, there's less down the axon. We've actually determined that by examining the biopsies and the proximal portion of the helix. There's a lower mitochondrial density. And there's this perinuclear clustering of mitochondria, but a lot of it is mitochondrial junk, partially degraded products. And this changes the balance. Now, one of the major things of this portion is that I've really highlighted a key role for amyloid in this process, and that seems to contradict all my scathing comments about the amyloid cascade hypothesis. So that'll be my next part, talking about that there's kind of a third ground for amyloid. One is that it causes everything. Number 2, it's irrelevant. But number 3, it plays a critical role, but it isn't causative. And that's the one I will take from this talk. So we wanted to understand, with this important role of amyloid for mitochondria and other things we had examined, what is it really doing in Alzheimer's disease? We had done culture studies that we had published many years ago, in which we found amyloid was not particularly toxic. In fact, neurons responded in interesting ways. And when we did oxidative stress studies in quantitative ways, we found that, if anything, there was a reduction in oxidative stress always around amyloid when we looked at human brain. So here I'm doing this study in Down syndrome. Down syndrome, the people invariably develop Alzheimer changes in their brain, always, in their teenage years, and have what looks like Alzheimer disease in their 20s and 30s. Their cognitive loss is later. People with Down syndrome do become demented in their later form of their life. So we looked at how oxidative damage in this series. So it's driven, everybody changes the same. That's different than normal people, that's very heterogeneous. So we looked at teenagers, and this is stained for oxidative changes, and you can see these neurons are pretty prominently stained. When we looked at older people, and 61 years old is old for somebody with Down syndrome, you can see here that the neurons aren't even detectable. And yet there's much more amyloid. That's the brown stain here. And we quantitated this and did it in decades. So when people are very young, 0 to 9 years old, they have very low amyloid, essentially 0, and they have very low oxidative damage, although it's above what you'd expect for a normal individual. But you see the oxidative stress goes up, and then at advanced age, in this case over 30, it's down. So they're actually temporarily separated from each other by at least 10 years. And if you correlate the two things, the amount of amyloid in the brain, amyloid burden, versus the amount of oxidative damage, there's an inverse relationship with about R² of about 0.95, which I thought was pretty amazing considering all the cases came from different centers and were handled differently prior to us doing them all the same. The other piece is that when we looked at when does oxidative damage occur for the average Alzheimer individual. So for Down syndrome, it looks like it occurs at least a decade before amyloid really starts getting deposited. This was a study we did published a couple years ago looking at cases that came from Washington University where they looked at incident cases. That means that they examined normal people through their life when they were aged, and they collected their brains and they knew their cognition. So there was people that stayed normal and died. There are people that had really mild changes that probably a family member would barely notice. Then there was mild cognitive function problems, and then mild Alzheimer's disease. And they provide those definitions because they're pretty subtle. But if you look at oxidative damage, again, is that the highest level was during this MCI. It's borderline statistical, but it was higher there. And then if you look around the body, like in this case, the controls were Purkinje cells of the cerebellum that are much less affected. You see there's almost no difference. Well, there is no difference statistically. So it's specific to the large pyramidal neurons, and the highest levels appear to be here, kind of recapitulating what I showed for Down syndrome. This is actually the first piece of data, not the first ones we published, and that is for sporadic Alzheimer's disease collected at Case Western, so went to a tertiary care hospital. And when we looked here among people, and I have them classified between ApoE genotype, the people with Alzheimer's disease, the higher the amyloid burden, the lower the amount of oxidative stress. You see among the controls, there are normal people with high levels of amyloid overlapping the Alzheimer one. There's no relationship of oxidative stress to them. And just like in Down syndrome, this curve is a curve of duration of disease. When people develop Alzheimer's disease, their death can be at any time. Older people have lots of health problems. They have other reasons to die. So some get a diagnosis and die within, a year or two. And some live, well, almost to 20 years. The average is more like in this range, 8 to 10 years old, 10 years a period. But anyway, a slight decrease. You notice through the aficionados that there's a difference with ApoE genotype because people with the E4 gene tend to have more amyloid deposits as the disease progresses. So on the same pattern, we looked at people that have genetic cause of having Alzheimer's disease, presenilin mutations predominantly, and we found the same thing. We found this inverse relationship, significant, not in the same as the other. The R-squareds aren't 0.9 anymore. And that relationship is for amyloid ending at 42 residues for the amyloid aficionados. All of the correlations are for amyloid ending at 42. If it's shorter, there's no correlation, as shown here, and that would be true for everything. In this case, we also had a pre-symptomatic N of 1, a person who died of a heart attack that was genetically, would have developed the disease, and had high levels of oxidative damage, but didn't have dementia or very much pathology in their brain. That's why there's no bar there. All suggesting oxidative stress is an early event during the disease, and if anything, is met by amyloid deposition by reduced oxidative damage. So why would amyloid, so this led Mark Smith and Aki Nandamura and myself to say maybe amyloid therapeutics won't work. So first one of these we wrote was called Amyloid Junkies. And saying, how do you know it's going to work? Maybe it's going to actually make people worse because this is a reaction to the thing. So then we tried to understanding, well, if amyloid had a protective response, what would be its mechanism? So we did this study, this is one of many, with Raman spectroscopy. We looked at isolated plaques. So plaques, as opposed to tangles, are pretty easy to isolate because they're defined structures. You homogenize the brain, you put it over a couple sucrose gradients, you get something pretty enriched, you run it through a cell sorter, and you have something that's almost perfectly pure, and it looks like this. This is stained with Congo red, this is stained with antibodies to amyloid beta. In this particular case, we worked with Paul Carey in biochemistry case, and he did the analysis of these. I'm not an expert at Raman. Is that we wanted to look a copper and iron binding. I was convinced there'd be both. But the only thing he found were these little blips, which he told me were clear copper binding in amyloid beta, okay? And zinc binding. So why would that be important? We'll come back to that in a short while. So more recent studies we did in San Antonio, we isolated plaques, which are shown here, and a couple different methods. And this is a scanning picture of one. This one particularly has a blood vessel, not all of them do. And you can see they look like little pom-poms you'd see at the football game, the cheerleaders or others. And you can take those guys and you can embed them in plastic and cut sections through them. And you can look at those. And why are we showing something which is just routine electron microscopy? Because when we had been looking at this, we wanted to understand where iron and copper were. They were there. Number one, I showed the spectra, and they only showed copper, and I was always convinced there was a sensitivity issue. So when we did this, we did something different with this electron microscopy because we didn't stain the sections. We didn't use urinal or lead salts. That's what you almost always do. When you're looking at electron micrographs, you're not looking at proteins, you're looking at lead and uranium or osmium. We didn't do any of that stuff. So this is being looked at directly, and you see these little white spots that you definitely would ignore, okay? Or I did anyway. So those little guys turn out to be magnetite crystals, pure magnetite crystals. So this microscope has 0.6 angstrom resolution. So we were able to show that they had these crystals, and they are pure magnetite having exactly the right crystal structure. And we also went in, isn't shown here, we looked where the copper was. And the copper was on the filaments, and the iron was in the crystals. So that kind of surprised me. Further, we were able to show that these guys are definitely magnetic. And you could say, well, magnetite's always magnetic. Well, it isn't when you get to this size distribution. This size distribution's right at the border to have magnetic properties or not. And we had known, Mark Smith had determined this, that if you take plaques and tangles, when you isolate them and you put a magnet near them, it's only an experiment that Mark Smith would do. He started moving them around with a magnet. And it took us 20 years to figure out how to have a paper that didn't have us with our hand with a magnet on it. So this is that experiment. Because what does it mean that you can move it with a magnet? Does it mean it's magnetic? It turns out they're super paramagnetic. And these type of particles were known in the brain before. There's accumulations of magnetite crystals. But them accumulating within the plaques like this was new to us. Okay, so we're doing more work to try to understand why it's accumulating there. The other part we're using in coupling with this, and I'm showing the experiments that won't be relevant to that piece, we're using MALDI imaging, MALDI mass spectrometry imaging. And we think this is going to be really a critical tool to understand plaques and tangles. So here we have a hippocampal section. And we're able to map where all the amyloid fragments are within that. Not at super resolution, in fact, only about 10 micro resolution, but you can see how they're distributed. We could do the distribution for anything on it. We don't need to have an antibody to it. We actually don't need to know that much about anything to be able to produce these images. But one of the key things this will be is that we can now look at individual plaques if we isolate them and zap them with the laser and look at the composition of each one. And we're doing that right now. This is looking at composition. And then a study done at the same time, we looked, well, when amyloid beta binds copper, who cares? Well, one of the things copper does is it catalyzes the oxidation of things. Catalyzes the oxidation of iron and reduction of iron, but it also oxidizes things. It oxidizes vitamin C. It oxidizes lipids to form hydroxynonenal, one of the toxic aldehyde products, carbonyl products of lipid peroxidation. So if you have just plain copper and you add if you add copper to arachidonic acid, a polyunsaturated fatty acid, you'll have increased productions of this lipid. If you add amyloid to it, you'll have a reduction because, because it binds the copper and suppresses it from redox activity. So that's one mechanism it would have, a stoichiometric method to reduce oxidative stress. And this led to us writing even more outrageous. Mark and I couldn't stop writing outrageous articles after amyloid junkies. So we call this one, Amyloid is a life preserver to the brain. So the idea that neurons actually during Alzheimer's disease are protected by the amyloid response from oxidative stress, and that's a predominant driving feature. And removing this life preserver isn't going to make things better. And that's been borne out by a number of the clinical trials recently over the last decade that have only failed. So last pieces, I'll talk about something that we found was the issue of cell cycle reentry. These neurons that are insulted by oxidative stress die, but how they die is really still very, very unclear. And when they die during the course of the disease, there's no correlation of neuronal loss to cognitive loss. And the shrunken brain I usually show and Alzheimer's disease are not always the picture of the way it occurs. So you have this two things, cell cycle abnormalities, and the one that's the best studied is this one. When cells get out of control, and that happens because you have defects like in children, genetic defects, or getting old and age-related cancers. Cells over-proliferate, and that's something that is bad. In Alzheimer's disease, it looks like you have some other type of problem, that cells are more quiescent. And this is a summary that Ahrens in Germany put together from our work and others, that every form of cell cycle reentry proteins is induced during Alzheimer's disease. And there's some indication from Carl Harrop and others that the cell may have actually replicated the DNA. We had some work that they replicate nuclei. You can see by nucleate abnormalities. But to address, is this really an important thing? So to address this, we looked at an animal model in which we forced the cells to go into the cell. I mean, we really hit it with a sledgehammer. We put c-Myc in, and we have an inducible marker, and it's particularly focused on expression in cerebral cortex only. And when we induce this thing, the c-Myc on, we actually have proliferation of the nuclear of the DNA. You can see DNA incorporation of uridine. Let's see. There's DNA damage marked by, and more importantly, there's definite cell loss. The cells die compared to when it's off. It's CAM kinase is cell loss and by apoptosis-like changing. There's gliosis, and I didn't show it, but if you looked at behaviorally, there's an effect on cognitive parts of the mice, but there isn't an effect on motor function because it doesn't driving the Purkinje cells. So it means that cell cycle reentry can be important in causing neuronal death. And the last piece that I'll go over really rapidly is about oligomerization. I know it's something people are studying here. And I've taken pictures that I didn't produce. They were actually produced by my mentors way before I worked with them. So these are pictures taken originally from the 1970s. And I would say even predate that work from the early 70s. So amyloid and tau have a lot of similarities to the normal cytoskeleton. You have monomers, and they eventually form polymers. So what do we know from the normal cytoskeleton? So if you take this case, I'm going to focus on tubulin, experiments done by Joe Bryan. If you look at the normal assembly, you have a pure solution of tubulin together with associated proteins like tau and MAP2, and you heat it up in a tube and you do EM of it. And what do you see? You see these little ring-like structures forming. You see all this stuff. They don't look like microtubules. And you form more like globby stuff, and you form more like sheety stuff. And if you wait around a little bit longer, you start to see something looking more like filaments. And here we have a cartoon, which is what everybody wants to have, okay, to simplify things. You start out with some little monomers, which in the case of tubulin are dimers, and they form little ring-like structures, and they form these guys, and they start to form. But once they start to look like a microtubule, you don't have any of these guys there because they add here. This is the energy minimum to add to, not these guys. So those of you that are oligomers and amyloid, you'll appreciate that difference. And then you look at a cell. How are microtubules organized in a cell? Are they ring-like structures? Wow, they're pretty elaborate. This is the organization within a fibroblast of microtubules. And I'll skip these things. Those are about assembly characteristics. You need extra proteins. The proteins by themselves, tubulin, has a very hard time forming filaments. Opposite. And let's go back to a cell. You can take a cell, you can get rid of the microtubules that are in it, and you can make holes in the membrane, and you can pour tubulin on top of it. And the tubulin will go back and stick onto the cell. And where does it stick? Does it form rings? No. The first thing it does, it forms little dots. And those little dots even become brighter, and they start to grow out. Those are the centrioles. So in other words, the tubulin even forms where it normally would have formed. It doesn't form something as nice as in a cell, you know, filling, kind of like radiates all over. So we used those experiments to try to understand how plaques were formed. And we did it by looking at labeled amyloid beta, and we did it amyloid tau. And what we found was something kind of surprising. We found that tau protein specifically bound to plaques. So this is bitinylated tau binding on tissue of Alzheimer's disease, and it's binding to where the plaques are. And what we found was it bound to a region when we mapped it out with using synthetic peptides and other things. We found that it had nanomolar affinity for a minimal region, just a little C-terminal to amyloid beta. This is affinity, and that shows that that sequence is even there. And the main point about that is that amyloid and tau forming polymers in the brain, forming organized structures, is probably very relevant, the prior structures that are always there, just like in the cytoskeleton. They're not random configurations. Understanding the oligomerization is important because it's easier to study. You can mathematically model, and that's what was done in the cytoskeleton, studying it during the 1970s. So I'm hoping that others, not me particularly, will try modeling this in a more cellular way. So what I presented today is oxidative damage is an early feature of Alzheimer's disease. And why I emphasize that is if you see the amyloid cascade hypothesis, they show oxidative stress somewhere in the basement as a terminal phase of the disease. And everything we see is that during the most terminal phase of the disease, oxidative stress is already gone. Mitochondria are critical to oxidative abnormalities. Although we don't know the complete connection, my own work, for me, suggests that that they are critical in producing the primary oxidant, which is superoxide, as well as leading the metals. And we know the two are correlated in data I didn't present today. Amyloid and amyloid precursor protein are very linked to mitochondrial turnover. The stuff I showed about fusion-fission being influenced by it. Other work that others have done have shown that amyloid toxicity depends on mitochondria. If you have cells without mitochondria, amyloid will have no effect on them. Copper and iron abnormalities are intimately associated with plaques and tangles. Well, they're intimately associated because they're full of them. Both of them have it. Nucleated assembly is important. In other words, the idea that you can have assembly in a homogeneous system, which is what you do if you put amyloid beta in a tube, may not recapitulate everything you're seeing in the disease. And cell cycle entry may play a direct role in neuronal death. The fact that neurons appear to be going into the cell cycle may play an important role for their dysfunction, but we need to understand a lot more. What are all of the features that are going? And the mimicked model only shows you what happens when you hit things over the head with a sledgehammer, as opposed to a mild change that happens during the disease. So I thank you very much for the attention, and really a great honor to be here.
Questions for Dr. Perry?
Yeah, I have a question related to your cell cycle hypothesis. So, and also linked to the oxidative damage. So in that summary where you show the expression of different cell cycle proteins,
Yes.
I couldn't avoid noticing that you do see overexpression of cyclin and CDKs, but you also see overexpression of CDK inhibitors, which
I know, you see everything.
so I mean, from the balance, I mean, to me that means that yeah, sure, you have your cyclin CDK complex, but it's inhibited and is present but cannot do absolutely anything. So
Yeah, it is not clear that they have entered. In fact, for our own experiments, we never saw that the neurons went all the way around. Actually, the only one who's reported that is Carl Harab, because he said that there was DNA synthesis and replication, and we never were able to establish that.
that makes more sense to me.
Well, thank you. Thank you. But I'm just saying that was with other pieces. We were not able to, it looked to us that cells were going into the entry point, and that entry point, but they never really complete.
And one more thing is regarding the oxidative damage. It's actually quite interesting. And I'm not an expert in neurons and much less on Alzheimer disease, but so usually what we see in cancer is you have an exacerbation of oxidative damage in the cell in general, and coming from very many processes undergoing really, really wrong, which end up in tremendous DNA damage, and usually that turn out into lack of cell death, which is what you see here, and uncontrolled proliferation and things like that. It puzzles me that the cell doesn't die.
It is really, I mean, the cells, look, here from about the age of 40 on, our brains show many of these type changes, even normal people. So the only normal people here are the students. Okay. Well, maybe not all of them, not all of them. So anyway, that our brain is inundated with all these things that you would think under all the models that are there should have led to cell death. If you have mitochondrial products in a cell, if cytochrome C release usually sets a cell up for death.
Yeah.
So I didn't look at cytochrome C, but cytochrome oxidase is even a more integral membrane protein, and it's in the cytoplasm. So is DNA. And this is, although I showed Alzheimer disease where it's really striking, if we looked at the average 50 or 60-year-old's brain, we would see some of the same phenomena, just to a lesser degree. How do cells live that way? Then when you develop Alzheimer disease, studies of Paul Coleman, or just looking with your eye, so to speak, neurons live for 20 years when people have Alzheimer's disease.
So my thing would be there then to think that it's an autophagy issue, that the cells, they don't actually die because the whole autophagy mechanism is somehow balancing.
I think what has happened is it's balanced in a different way than you would expect. That you've recalibrated. I mean, that's why I mentioned about the oxidative stress idea, because if you think of the old idea, everything should just go to crap. It should just go down the hill, rust away, and you'd be dead. That's where the 1960s and 70s ideas were. And instead, this system appears to be for chronic conditions, hit some new equilibrium, which is stable, not necessarily good, and we don't really understand it particularly well. So this accumulation, we talked about fusion, fission. There could be the transport phenomena, even though we were not able to replicate that changing microtubules that much. We did a more recent studies to actually look what type of microtubules are left. It's not the same. There's not just one population of microtubules. Microtubules exist in at least two populations, what's called stable microtubules and the unstable. The ones that are left are the stables. And there's a biochemical marker for them. They're more acetylated, tend to be more acetylated, and that's the type that tend to be left. And we don't know that microtubules are limiting. In other words, even if you get rid of 80% of them, maybe it doesn't make that much difference. Back to the other piece is the autophagy piece, is a degradation problem. In other words, you're actually degrading the same amount of mitochondria, but they just don't get chewed up as much. So my own idea right now is that piece of degradation and how it turns over is really a key part of what we're seeing in the pathology. Because when you have a problem of accumulating mitochondrial degradation products, a key part of those is a bunch of metals. And how do you deal with those metals? There isn't, and I should add this, there's actually no increase in metals during Alzheimer's disease. Despite seeing, and I didn't show here, if I showed you metal activities, like able to do redox chemistry, there's a big change, but total metals don't change. And there's even a new paper, a meta-analysis of oxidative damage. And there may be little change in oxidative damage. It's more about temporal changes and specific cell compartment changes.
I'm interested in the metal topic. Has anybody measured the affinity of iron or copper to A-beta?
Yes. Craig Atwood, who used to work with me before he worked with me, did that type of work at Harvard. They said it was femtomolar. They said it was equivalent to superoxide dismutase affinity. We've had a problem really addressing this in a very exact way. Because what I'm trying to do right now is to understand if when the copper is bound, it isn't just that it's being stoichiometrically tied up, that it's actually becoming ferroxidase. That's a more esoteric thing than even normal oxidative stress. So copper and iron usually control each other in one way or another. So the classic one of them is a protein called ceruloplasmin, which is in the bloodstream and is an inflammatory protein and plays an important role in controlling iron redox. And so in my own brain, not my pathology, but my thought is that is what amyloid beta is playing. And that activity of ferroxidase is what ferritin does too. So it's important to have iron change from iron 2 to iron 3 in a controlled fashion. And copper is an important catalyzer of that. And I think I may have published that in the past, but there's several weaknesses of what pieces that are missing. And it's not so direct, not so easy to directly show it. I know that's an ambiguous question because we showed that tangles and plaques have ferroxidase activity. We published in the mid-90s. We thought it was iron that was present in 97. And iron also binds to amyloid. So I was convinced iron and copper would both be there. And the answer is there's low amounts of iron present. We have another study that will be published shortly with using the synchrotron radiation, looking for metals, which has, I think it's complementary to what I presented. It doesn't have as high a resolution, but it has some other metals we can spot. And definitely you can find some degree of iron there, but copper is the dominant thing when it's at the filamentous level, and then these crystals. So the idea I have is the crystals are produced from the fibers. Oh, yeah.
It's so relative, so the affinity is really what is essential because especially, iron is not really freely available, so you have to have a very high affinity to actually find it in vivo.
So that is, when you do this, if you do amyloid synthetic peptide, you can get almost any metal to bind to it. Yeah. So we're doing these with isolated plaques to see what actually was picked up in the brain. And there is some downside with that because we isolate them, so they could have fallen off and it could have been there, but we haven't got to that point yet about worrying about the come-off state. And actually what we're doing right now is using the imaging mass spectrometry to look. Copper binds to amyloid and changes its spectra on mass spectrometry. We have a paper in, I think essentially in Press on this topic, but that is not to look at the physiology. It's where we pour a whole bunch of copper on and we look at the spectral shift because it actually helps with, as a method for mass spectrometry. To look at the endogenous copper, we haven't seen a clear spectra. And there's a couple reasons this could be the case. One is that amyloid beta really doesn't have copper on it in vivo because the Raman spectroscopy really didn't say that the stuff was bound to amyloid. It just said it was in plaques and on histidines. That's all that showed. At the point we did this, we thought plaques were pure amyloid. They are not pure amyloid at all. They're full of other proteins, and that we're publishing because, when you look at these things, like the things I showed you, they look really pretty pure. They're filaments. They look the right dimensions. You don't see any other junk. So why wouldn't you expect to have spectra that just show amyloid beta and amyloid beta fragments? That's not what you see. You see a bunch of other things. Are those other components binding copper? I don't know. I tend to think it's the amyloid based on other people's experiments. But I would say the direct evidence we have, we only know it's present in fibers that are not pure. And if you're talking about tau and tangles are much more complicated than this thing.
So you had done a study, if I understood correctly, this was with some of the multi-mass spec imaging with some hippocampal tissue, and it looked to me like you were expressing A-beta fragments of different lengths.
Yes.
What were your conclusions from that study?
Oh, this was mostly done not to even address that question, okay? It was mostly addressed that we could do it. We did 2 studies like this. We wanted to show that we could actually use this method reliably in human tissue. So we picked something that was, pretty easy we could have synthetic peptides of, and that we could be able to identify all the fragments of. And the conclusion otherwise is that there are many different fragments present in plaques and in the brain. It isn't just 40 to 42. That isn't a new finding. But it's nice that you can have that fragment identification with some anatomic coordinates in tissue. And that's really what it was. It was more about methods development. When we're actually using it right now, we're not using it in tissue. We're using it in isolated structure. So why do we need having this thing? Because we're really concerned about each plaque and tangle being real. When you have a whole bunch of these guys around, you're not sure that they're all real. There's other things like blood vessels get in the thing. In the case of tangles, they're not a defined structure, so I can't isolate them by the cell sorter. It's not as reliable, although there are new cell sorters, so it might be better now than it was. So it's really useful to be able to get composition on single structures and see if they all have the same spectra, or 90% have the same spectra and 10% you don't know. So it's really methods development. I'm interested in knowing them for a lot of different structures. I'm mainly focusing on plaques because plaques I can isolate very pure, 95, 99% pure. So I could take a whole population and be fine with it. When I get to tangles, it's not. So we've already done spectra and found that we could get tau and fragments of that. I'm interested in other fragments, like whether tangles have lipids and other things. So no one has done human tissue of Alzheimer's disease before. Those are the first studies. There had been some studies of mice that were genetically modified.
Before we conclude, I will make a quick announcement. At 3 o'clock, if anyone's interested in a question, want to talk to Dr. Perry, we will have a live reception just at the Freeman kitchen. And also the student who is on the list for lunch, please stay. And lastly, we have a VT colored present for Dr. Perry.
Wow, wonderful.
Thank you.
Oh, wow. Thank you. And you see I have half the colors. That's hard.
I wanted to ask you a quick question. I don't know if it's really a question or not, so I didn't want to say it in the room. So we've been doing some work with herpes simplex virus.
Yes.
And our normal model is adult sensory and autonomic neurons, and we had a collaborator contact me and say, can you test this in hippocampal neurons, adult hippocampal neurons? He had some TK inhibitors that he wanted to try to see if they'd work. And so I had absolutely no information about Alzheimer's, never worked with Alzheimer's.
There's a lot of people who think herpes simplex.
All right. Yes, and that's what this guy thought. And it doesn't matter.
Okay.
So, so he's a kinase inhibitor guy.
Okay, so he's not.
No, and not an Alzheimer's person at all. And so we had this sort of